So today we are going to finish chapter 14. I told you guys it was a very short chapter, and yeah, I wasn't kidding. So today we're going to go over the first life forms. Now, let's go back a few chapters. So when we were making a protein, we went from DNA to RNA, mRNA to be specific, through transcription. And then we took that mRNA, we left the nucleus, and we turned it into a protein. In other words, a bunch of amino acids bonded together. Now, scientists are wondering now, okay, why did we need that RNA? Why can't we just go from DNA to an amino acid? Okay. That would be a lot simpler, right? So we found that, and we'll go over that in a minute or two, guys, but we found that in our RNAs, some of our RNAs, like tRNA, if you guys remember, it had like that clover shape to it. I'll draw it for you here real quick. If you remember, we had our tRNA that brought by the amino acid, and that tRNA kind of looked like a clover-looking guy here. And in these three spaces here, we had our anticodon, which I'll just make a easy one here real quick. We'll just do, I don't know, A, U, A. Okay? And on the other side of it right here is where our amino acid is carried. But all of this stuff in here, guys, all right, all of that is also our RNA nucleotides. So the three nucleotides right here is not the only area that nucleotides are found in our tRNA. It's all throughout the outside as well. But if you notice, still, it has this weird looking clover shape to it. Well, what causes that clover to stay together? Why isn't it like a long string? Well, why it's not just a long string of nucleotides for the tRNA is because we have hydrogen bonds in between our nucleotides. So again, we have our amino acid right here. We have our clover looking guy here. I think we did AUA on the last slide. But what we'll have here is we will have some hydrogen bonds in between these ones here, okay, making them have some type of shape. So it's not just like one long chain of nucleotides in a circular form. There actually is hydrogen bond interactions between all of these nucleotides together. So here is our string of amino acids, and you guys can see here kind of where the S's are. We can have some hydrogen bonds just like we do in making a protein, okay? When a protein is made, after we have all these amino acids, it folds up. We can actually kind of do the same thing in RNA. Okay? So what we were thinking was that this RNA, given that it can fold up and form hydrogen bonds in itself, maybe it can act like a protein at some time. And if you guys remember, what is a catalyst or catalyze, when we catalyze something. If you remember, when we had enzymes were just catalysts that are in living things. They helped us to lower the amount of activation energy or energy needed for a reaction to occur. All right, so that was a catalyst, if you remember from the past. So a bit after we're starting to study all this stuff, this is in 1980, so really, not that long ago, 40 years ago. A scientist named Thomas Cech confirmed that there is an RNA that's found in eukaryotes, and it's able to actually act like a catalyst. It's able to act like an enzyme, okay, which again is a type of protein. He called this a ribozyme. Okay. So why, again, going back from the beginning, why do we need this RNA? Well, if we if we went from DNA to a protein, like I said at the beginning of this lesson, it would eliminate a whole step for us. So why do we even deal with RNA? Here's why we need it, guys. if you remember, for DNA replication to occur, we have to have an RNA primer, and if that RNA primer is not there, DNA polymerase can actually not bond to that area, and then we can't have our new strain of DNA. So in other words, all cell reproduction would actually halt, which, yeah, not good. This also tells us that RNA was actually the first genetic material out there. It came before DNA. Because we need that primer there for DNA polymerase to latch onto, if there's no primer, we don't get new DNA. All right, so if RNA wasn't there first, we would have never had DNA because we need RNA to make DNA. So we think that early Earth life was actually made of RNA. Another hypothesis is that, and keep in mind, guys, we talked about microspheres and coacervates, which are pretty much like empty cell membranes. They had to compete. So once they had the RNA inside of them, like the RNA chain, in other words, the backbone of RNA, they had to compete with other RNAs in order to make a full RNA molecule. In other words, we had an RNA strand, just the backbone, so it would have been composed of phosphates along with ribose, but it would have been missing the A, the U, the G, and the C. So it was competing with other RNAs to try to collect these nucleotides, collect the A's, the U's, the G's, and the C's, until it had a full strain. Some of them were in a good sequence, some of them were not in a good sequence. All right? If they were not in a good sequence, they probably died. If they were in a good sequence, they survived and made more cells, early cells, obviously. Okay. So Cech later found that these ribozymes, which again, guys, are RNA, they could actually replicate themselves. So if they were able to replicate themselves, now we had more of these nucleic acids, these RNAs on Earth, and we had more early life forms, I guess we could say. So they weren't really that complex, guys. It was pretty much just, our nucleotides bonded together. Okay, so in our first cells, guys, our goal was to make a cell membrane with some nucleic acids inside, all right, that was able to function. So we think that at the beginning of Earth, there was little to no oxygen. And our oldest fossils that we can find actually look like prokaryotes. Now, obviously, they would have had RNA instead of DNA, but they did not have a nucleus. They did not have any organelles. So the first cells that we found, given that there was little to no oxygen on Earth, they were probably anaerobic. So anaerobic, they don't breathe air. Okay. They also were heterotrophic, meaning they could not make their own food. So what happened here, guys, is our early cells had to compete for food. Some got the food, some did not. If they got the food, they survived. If they didn't get the food, they died. So in our first cells, guys, we think that the ones we were studying kind of look like archaea. Again, it's a prokaryotic cell. Archaea lives in harsh environments. And we think early Earth probably was a harsh environment. So after this, we started to get different species out there. The first one is archaebacteria. It's a unicellular bacteria, and it goes through a process called chemosynthesis. A little bit different than photosynthesis. So photosynthesis, if you guys remember, photosynthesis is going to be an organism that is going to take in carbon dioxide. It's going to be able to make its own sugar. All right. Chemosynthesis is a little bit different. Still, carbon dioxide is serving as the carbon source, and it's going to assemble organic molecules. But our difference between them is chemosynthesis. They are going to use carbon dioxide. They're going to assemble their organic compounds solely out of that, where photosynthesis, if you guys remember, they need sunlight in order to assemble their glucose at the end. Chemosynthesis, no sunlight. After this, so about 3 billion years ago, we started to have life that could become photosynthetic. This means that they are going to go through photosynthesis. So before this, it was probably about 3.5 billion years ago. So 3 billion years ago, we started to get our photosynthesizers. What this meant was if an organism was going through photosynthesis, we were starting to up those levels of oxygen in the environment. Our early prokaryotic, photosynthetic, unicellular organisms were called cyanobacteria. All right, cyanobacteria is still here today. Now, it was a little bit different, obviously 3 billion years ago, but it really closely resembles cyanobacteria today. So cyanobacteria, photosynthetic, unicellular, prokaryote, all right, and that was our first photosynthesizer. So like I said, guys, we started to get oxygen. With oxygen, we started to have a mass extinction of our organisms in the world because some of them, they couldn't tolerate the oxygen being there. So that was our first big mass extinction. So what we think, guys, is it took about 1 billion years. So this started 3 billion years ago. Oxygen levels didn't reach to where they are today until 2 billion years ago. It took 1 billion years for oxygen levels to get to where they were. You guys would think, okay, photosynthesis, yeah, they're probably producing a good bit of oxygen, but there was none there originally, and we didn't have a whole lot of different species that were photosynthesizers. That's why it probably took about 1 billion years. Now, oxygen that we know it, guys, as we know it, the stuff we breathe in, it is the chemical symbol O2. So it's 2 oxygen molecules bonded together. That is oxygen that we breathe. That's O2. Once this oxygen from those photosynthesizers reached upper levels of our atmosphere, it became reactive. When it became reactive, it actually bonded together, forming O3, which is 3 oxygens bonded together. And that became our ozone layer. That helps us from all the harmful things of the sun. Now, obviously, some of it still gets through, which is why we can get sunburn and stuff like that. But it does protect us from a lot of the harmful stuff from the sun. And our atmosphere is actually kind of healing itself right now a little bit, our ozone layer, just because of the whole pandemic. We have been more inside than we've ever been in the past. So here's our ozone layer, guys, right there. And like I said, that's going to absorb a lot of our harmful rays. Okay, so now we're going to talk about the rise of eukaryotes. So far we started just talking about prokaryotic cells. So Lynn Margulis, she proposed that eukaryotes evolve by early eukaryotes engulfing aerobic prokaryotes. So we had these prokaryotes that breathed pretty much. And they were just kind of chilling there. They could live on their own, but they weren't having the best life. So what happened here, guys, is we think that our early eukaryotes, where we started to have some of our organelles, they actually engulfed some of these prokaryotes. In other words, they ate them. And it wasn't like they ate them to the point where it's like, oh, I'm going to eat you, I'm going to chew you. No, they didn't. Like, cells don't have teeth, guys. There are teeth cells, but you can't think of these unicellular organisms as having body parts like we have. So we think that the eukaryotic cells engulfed them, and why they did this was so both of them can benefit from this engulfing. If you remember, we talked about endosymbiosis a while ago in the endosymbiotic theory. which stated that the mitochondria for us, for animal cells, was engulfed. And for plant cells, it was the mitochondria and the chloroplast, both of them. Okay? And the chloroplast goes through photosynthesis, if you remember, which is why plants have the chloroplast. But both of these were prokaryotic cells, the mitochondria and the chloroplast. What we were providing, the eukaryotic cells, we were providing a home for them, so we would take care of them. At the same time, the mitochondria and the chloroplast helped our cells, helped eukaryotic cells with energy, like making energy and using that energy. So, in other words, they both dealt with making ATP. All right, and here's the one we already talked about, guys. It was the mitochondria and the chloroplast. Why we think it was these two was because they have their own DNA, and they still do to this day. They really closely resemble prokaryotic cells with respect to their membranes, and they don't have organelles. Okay, they do have ribozymes, but that's pretty much it. So, like I said, we gave them a nice stable environment, and they gave us the ability to make energy for ourselves. Okay, so that is the end of chapter 14, guys. I told you it was going to be a very short chapter. So we'll review, and then we'll have our test on this. Have a good rest of your day, guys.